will be able to see my screen. So this is kind of a small session, small structural biology presentation, structural bioinformatics from sequence to the structure of the protein. And it's part of, I'm very much thankful to ISCB for giving me this opportunity to present this to you guys. So learning objectives for our today's tutorial would be to how to retrieve protein sequence and how to analyze it. Then I will introduce you to the micromolecule structural database, how we can use that, how many structures are there, and how we can download and visualize those structures. Learning about the structure visualization tool. So we'll be using one specific tool for structure visualization and analysis. And finally, we'll predict a structure of a protein whose structure has not been available through experimental studies, for example, X-rays or NMR or electron microscopy. So we will be building a homology model. So this is our question for today. This is so for this tutorial would be around this question. So we have this sequence one. This protein is a thermostable protease that the detergent company sells under to the washing powder industry under the trade name of 7As. And here I have put in some information which I will later on discuss with you guys. And then the second protein, which whose sequence, who is also a thermostable alkaline protease, but it's from a different bacterial species, and it's kind of a quite cheap alternate for the 7As. So our question today is whether the enzyme 2 can be a cost effective alternate. So yes, you can say, okay, you can go to the lab and perform some experiments, but that will cost you money, time, and everything. So before going there, we can perform some structural and sequence analysis by sitting on the table, and it will give us, and on our laptops, it will give us some idea whether it's worth going to the lab and perform more experiments, or we should just stop here. So yes, and that's what we are going to do today. So we're going to use computational tools to answer this question, whether another enzyme, which is a protease for sure, but from a different source, can it be a cost effective alternate for the 7As. So for this, what we are going to do is, and this would be a hands-on activity. So I'll be performing everything with you guys on my screen, and you can copy that and follow. And if you have any question, you can ask that as well. So we will first go to the UniProt KV, which is a protein database, sequence database, and it has a lot of other information available over there. So we'll go there, we'll check the information regarding protein sequence, its structure, what kind of function it has. And then secondly, then we'll go to the PDB, which is Protein Data Bank, where the structures are available. So we'll see if any one of these proteins has its sequence available, structure available, and we'll download that and we will analyze the sequence as well, a sequence and structure both by doing this. And for the visualization of the structure, we will be using a tool which is PyMol. The PyMol is basically a free software for the educational use. It can be used for the structure visualization, and it can gives you, it's a very handy kind of a tool, and it's available for Windows, for Mac, and for Linux systems. And finally, we will be building homology model, as I already know that one of the sequence enzyme 2, its structure is not available. So we will be using Swiss modeler to build the homology based model of this protein. All right, so this is it. Then so I just want to introduce here what homology modeling is. So homology modeling allows you to build the structure of the protein where you only have a sequence available, and you can use another template, another protein which is similar to your protein and whose structure is available. So I'll come back to this bit later on in a little bit more detail, and then we'll discuss more about homology modeling. But let's just get started with our hands-on session from the protein sequence databases, and how we can use that to get some valuable information. So here we are back, and all right. So now what we have to do, we have to we have to use UniProt uh, KB for the protein data search. So I'll just open a new web log here. All right. So I'll be opening my, let me just close these. And so you can, UniProt KB. Uh, you can, but it's journal to take you. It will, the first link would be for the UniProt KB, and here you can use the session number. Okay, what is an session number? So in my presentation, I have put in the session numbers here. Yeah, look at this. So sequence 1 have this session number, and the sequence 2 has this specific session number. The session number is a unique number which these protein sequence and even genomic databases gave to each sequence. It's a unique identifier. So the P29600 is a unique identifier for the 7A sequence, or all the information related to the 7A sequence in UniProt KB would be under this session number. So it's a very, it's kind of a very unique identifier. So we can, by putting this number in, we will directly reach to the 7A's page where all the information is available. All right. So here we are. We'll just put this in and we'll press search button or just enter. So here we are on the Savinase page, Subtalis Savinase. So this page has all the information regarding this enzyme. It has name, taxonomy, subcellular localization, phenotype, variants, post-translational processing, structure. So family and domain, sequence of the protein, and other similar proteins. So it has all the details available regarding this specific enzyme. So we'll go first, so you can go scroll down and you can actually go and see all the information which you are interested in. But first, I would like you guys to go to the feature viewer. So the feature viewer is basically a page which gives you information about the key features regarding domain sites, binding sites, active site, molecular processing if it any happens, structural features, whether there is a alpha helices or beta sheets present or turns present, then if there is any 3D structure is available and what's the coverage for that structure. So at the moment we are interested in here about the bind of the active site of the enzyme. So we can go, and so these dots are actually presenting the active site of the enzyme. If you use if you come here using your mouse, you can click on this one and it will give you the residue number, but it will also provide you a link to the ProSite. So ProSite is a website, is a database which has the information regarding the active sites of the proteins or binding sites of the protein. So let's just open that and look at more detail about the active site of this enzyme available on the ProSite. Okay. So here we are on the ProSite page for this the specific active site present in our enzyme. So it has this ProSite. This is also an identifier for the same for this enzyme on the ProSite subtalase, which is serine protease. It has a subtalase there. It's coming from this from the bacteria, which name is subtalase. So you can come down a little bit from the scroll it down and you can see here the active site residue. So it has a serine protease and it has this TH histidine and serine based active site. And these are the specific residue numbers which are given here. And but these are the three conserved residues for the specific serine protease which subtalis protease belong. So savinase enzyme basically, which is its commercial name, belongs to. All right. So you can note down these numbers and the residues, and we will later on discuss this on the we'll later on highlight this on the structure of the enzyme. All right. Okay. What else we can do? uh, Other than that, you can look at the structures and you can see whether any crystal structure available. So yes, there is a crystal structure available. So it's the structure is basically experimental structure, and there are multiple structures available. This enzyme is of quite commercial importance. That's why multiple structure with different modifications are available here. So just note down the PDB number. So this PDB number is a unique identifier for the structure of the this enzyme. For the and each one of these has a different identifier because each structure has been solved with a different modes, with a slight modification or by a different lab, and they have reposited it into the protein data bank. Okay. Remember, the session number we discussed earlier is a session number for the sequence of this protein in UniProt KB. So a session, and this number, this PDB identifier, is the identifier for the structure of this protein. All right. Then what else what so there are quite a lot of other information available, but we are not going into that detail. What we want right now, so we now know the active site of the enzyme, and we also know that its crystal structure is available, and now we're interested in its sequence. So you can, if you're interested in the sequence, you can go and download, you can go to the sequence page and you can download the sequence in the FASTA format and you can save it if you want for the later use. Okay, so I would just open in a new tab. I'll copy this sequence and I will save it in the notepad basically. Savinase, I'll name it as Savinase, our enzyme 1, and I'll just copy it and I'll save this file, save as, on the desktop under the name Savinase sequence. Right, here we are. So we have saved this sequence. Now, what else we want? Now we have another sequence, we have another protein whose in which we are interested in, which is our target protein, and we want to know about this, whether it's a serine protease, what kind of active site it has. So we have the session number for this protein available here. I have also highlighted it here. Copied it. I'll copy it from here and I will search it again in the UniProt KB. All right. If you don't have the session number, don't worry, you can search the protein by its name as well. And it will give you, UniProt KB will give you a list of the protein with the same name, and then with a little bit of focus you can see, but looking at the source of the protein, you can identify which one is the is your interest, is of your interest. So same as a Savinase, thermostable alkaline protease has a different source. It is coming from a different bacterial source, but all other information is available here regarding this protein. So in a similar way as we went for the Savinase, we will look at the domain features, feature viewer of this protein, and we want to look at the active site of this protein, whether it's a serine protease or not. So if we look at the active site of the protein, we'll go to the pro site and we'll see what kind of enzyme is this. So again, it's a serine protease. So it's taking us to the same active site. So pro site has the active sites database for all different enzymes. It has actually different domains or active sites saved in it. So look at the residues. So they are the exact same residues which are present in the Savinase enzyme. All right. So that gave us one answer, that yeah, one confidence that okay, so this second enzyme, which is a cost-effective alternate of Savinase, it is actually a serine protease and it has the same active site. Okay, so that is really hopeful. So that means we can carry on our study, and we and now we want to know more about the these proteins if we want to in terms of structure. So now we know the sequence has some similarity, at least the active site is very similar. All right, and now let's see if the domains, if the structure of this protein is available. So look at here. So this is a predicted structure. So the no experimental structure is available for this enzyme, our target enzyme thermocyclase, thermo stable alkaline phosphate or alkaline protease. All right, so that's we know. So we'll go and build the homology model for this protein later on. And in the similar way, you can go down and you can look at different details under all these feet in this on this page uh, regarding the type of post-translational modifications happening. And for example, the signal peptide is initial sequence is a signal peptide, it is removed, and later on you have the complete protein without this signal peptide. Similarly, you can go and look at the download the sequence of this protein. So I'll open the sequence in a new format. So this is alkaline phosphate sequence. Earlier we saved the sequence of 7As in the notepad. Let's save the sequence of the alkaline phosphatase, thermostable alkaline protease here. Save as it's thermostable alkaline protease. Okay, so we can save this at desktop. Okay, I'll minimize this window. All right, and you can go and look at different. If you're interested, you can look at other proteins which have similar kind of sequence available or similar kind of structures or properties. So a list of that would be provided here. Okay. All right. Okay. So now, so this UniProt KB is basically a protein sequence database, and but it is linked to other databases as well. So it is linked to protein database, protein PDB, which is protein structural database. It is linked to gene bank. So if you want to go for the genomic sequence of this enzyme, it will provide a link somewhere here where you can actually go and download the genomic sequence of this enzyme using gene bank ID for this protein. So these databases have basically link you to the various resources and provide you all the information. If you want to go in more detail, you can go to those web pages. For example, in our case, we actually used pro site, which is basically in which you have different active sites or different domain structures available, and you can go and check what kind of domain or motif structures are present in my protein of interest. So our both proteins have serine protease active site. So that's what we came to know from the pro site analysis. Okay. All right, now what our third step would really be. Okay, so we did. So this was our flow of information, flow of hands-on activity. So we have done this so far. Next, we want to go to the protein data bank. So here is a web link for this page. I can open this one, or you can just directly search this protein data bank in Google, and it will take you provide you the link for this page. Okay, so here we are. We'll just paste this one here, and now we are on this protein data bank page, and it has all latest publications, latest news or entries, and here is some information how many sequences are available. Some of the sequences are basically the predicted sequences or models, and some but rest of the sequences are basically the experimental structures solved using NMR or crystallography or electron microscopy. All right. So, do you remember in our when we were looking at PDB structure here, it provided us here a PDB ID for our subtalis protease enzyme, which is 7-ase enzyme. So I actually noted down that PDB ID for that enzyme. We can use this PDB ID to actually go to the structure of this 7-ase enzyme directly by putting it here, right? And we can search this one. Okay. So here we are. So here at the top we have our enzyme available. So you can open this one. By the way, you can also search the same structure by name, but then you have to look because different variant structures would be available. Then you have to go and see each structure and see which one is of your interest. Okay, so this is basically the page for our subtalis protease, and here is a publication which is related to this structure is given. It has given you how many chains it has. So it has only one chain, and then it also provides you any mutation. So there is one, no mutation has been done, but in other structure there might be some mutations available, and then some reference data is given here. What we can do here, you can download the structure in PDB format from this web page, or if you don't have structure, if you don't want to download the structure, you can even go click on this PDB on this page, and it can actually. All right, so here you can actually go to the viewer page, and you can visualize the structure on the virtual on the web page, on the same web page. So you can use this for the visualization of your structure as well. But of course, it doesn't give you much of freedom to actually analyze your structure. So this is your structure. Here it has given different binding sites, and other residues have been labeled, and rest of the details are given here. Secondary structure where the alpha helices are, where beta sheets are, have also been provided here, and you can actually have a look on your structure here as well. But what we are interested in here at this point, we are interested to actually visualize our structure using a tool which is called PyMol. And for this, I already have PyMol on my laptop, but I can teach you how you can download this tool and how you can actually install it. And so for that, you can actually search PyMol in the Google, or you can use this link to go directly on the home page of PyMol. And from that, you can actually go to the down. Here, some of the information is available, or you can go to the download pages, which is taking you at the end of this page. And depending upon your Windows or laptop specification, you can actually download the PyMol version, and it will ask you for the, and you can install it. Then it will ask you to register your actually PyMol, and you can actually register for the educational version. And there is another way to you can actually download the educational version. So at the moment, I suggest if you are doing it with me in this tutorial session, just download it and use it, and later on you can go for the registration. But if you have time, what you can do is you can go to Google and you can ask PyMol and download for education. Select this for educational use only. Okay, and here you can provide your information and continue. and then later on it will send you an email with the link of the PyMol, which you can use to download the registered version of PyMol, and you can actually use that. Okay, that's what I have done on my laptop. All right. So once you have downloaded PyMol on your computer, and you have actually you can actually use that. You can actually use that to visualize the structure. So in a bit, I can show you how we are going to use PyMol to visualize our structure. Okay. So here we are. So our structure has been downloaded here, and I already have PyMol on my laptop, so I can open this. It'll take a while. All right. So here we are. So this here, because I have downloaded the educational use version, so this has been written on my version that for educational use only. So here I will open my structure which I downloaded. So it must be in my downloads. Okay. So here we are. So this is our structure. Okay. So this is our structure. All right. So what are these? Oh, all right. Let me just hide this one. Okay, so this is our structure. This is our protein 7As. Right. So here in the display, you can go and select sequence. So it will display the sequence of the protein here. Okay, at the top. So it has only one chain. So here is a sequence from residue number 1 till residue number 271 or 276, I guess. And then it has some small molecules attached. For example, it has two calcium molecules attached and one sulfic oxide, SO4 molecule, sulfide oxide molecule attached. And then these are actually, the structures are usually bound to the water, either crystal structure or NMR structure. So these zeros basically represent the water molecules. So first what we'll do is you can actually select the, if you're a very beginner, you can actually, here you can use the command line as well. But because I'm expecting you to be like really beginner in this field, so instead of using the command line, let's use the mouse and we can select these water molecules, and we can actually here from the action. So the A stands for the action, and this is our selection. So we can name it like rename it. So let's rename it as water. Okay. So now our selection has saved as water. So this is our structure. Okay. And this is water residues. And A is for action, S is for show, and H is for hide here. And then L is for labeling, to label your protein, and then C is for different colors you want to choose. So I'll be using um, these um, these tabs for different actions. For example, I have selected the water residues and I want to hide them. So here you can go and you can, like, for example, hide everything. So now the water molecules are no more in the structure. All right. So because we are not very much interested in the water molecule at the moment. What else we can do? Because look at the calcium residues. So they are there, but they are in green and it's very difficult to look at them. So I want to color them differently. So I can select calcium residues and I can color them, like for example, I can color them orange. So now they are a bit more visible here. Okay, and this is our SO4 molecule. All right. What else I want to do in here is I want to, so by the left key, by the um right key, you can actually zoom in and zoom out this structure. And if all of a sudden your structure disappears, you can go here and you can actually zoom in. So it will actually give you your window in a your structure will come back actually, because sometimes it's just very difficult for the beginners to keep the window in a format where they can use and annotate their structure. For example, if something like this happens, you can actually go here, and in the actions you can actually press zoom, and everything will come back to normal. All right. So what now I want to look at the active site of this enzyme. So here, because I actually uh, read the paper or publication associated with this structure, so I know what the active site is. But we already know that serine proteases have T, and then they have a serine residue at 67 or something. Let me give me a moment. I'll find out where our other residues is. But the third one is at 221. Okay. And then we have this one, D or H. We want histidine. Yeah, we are looking at for the histidine, which is present at 67. So this is our active site residue. So we can say we have selected our active site D, H, and S, our active site residues. So we can actually rename it as active site here. And now our structure, our residues, these three residues have been saved as an active site. And what I want right now, I want to color them differently. For example, I want them to be red, and I want to show it as not. So this is kind of our cartoon image. For example, I want to show the side chains and everything, so I can go in here. So they are zoomed in right now, so I can show them as sticks or lines, or whatever you can choose. For example, I have chosen the sticks. So these are the our active site residues. We can look at them, and we can actually see their orientations and how they are available in the structure. Okay. So this is our structure. So this is how you can use PyMol to visualize and annotate the structures of your proteins. So let's just close this for a bit, and let's move on to our next step, which was all right. Some of the people are experiencing some issue. Uh, Yeah. So Sabaha actually asked, how do we identify the active site? So in the beginning of the tutorial, when we were looking at the when we were looking, I can actually go back for you very quickly here. So either you already know from the literature your active site, but if you search your protein on the on here over here in the on the Uniprot KB, you can actually go to this future viewer, and here you have the domain sites. So here they have given the active site residues have been highlighted in these small pink dots. So you can actually click on them, you can write down their residues numbers here from uh, well, in fact, it's more appropriate to actually go to the pro site, which has more detail regarding the active sites of this enzyme for all for different proteins and enzymes. For this enzyme, they have given a link of the pro site here. So if you click on this any one of these active site residues, it will take you to the ProSite web page, and there you can actually look and detail the active site residues. Okay, and in the structure, because when they solve the structure, they actually clone it. Sometimes the active site residue could be slightly may not match with the exact same number, but mostly they do match. And But for that, actually, it's more appropriate if you're going to the structure, you read the publication with that for the how they have solved the structure. And here they have also provided the, this is basically the domain. For the domain details, you can also go to the pro site for the link for the binding site residue. So this is a binding site residue. You see, there is a difference between the active site and the binding. So these are the binding site residues. They have also, the numbers have also been given here. So here you can actually go and look at the active site and binding sites, and you can identify which where the active sites and binding sites of your protein are present. Okay, I hope I have answered your question. Okay, all right. Okay, let's move on. And so, what we have done so far, we have used UniProt KB for the sequence visualization, for the identification of different features of our sequence, to look what kind of details are available over there. And then we have also downloaded the sequences of our proteins. And then we went to the protein data bank. There we actually downloaded the structure of our protein, and we used PyMol to basically visualize the structure of our protein and to actually highlight different features. For example, the active site, the bound, small residue, small molecules bound to our protein. So now we know how to use UniProt KB, how to use protein data bank, what is protein data bank, how we can download the structures from there, and what is PyMol, how we can use it, and how we can actually download and install it on our personal PCs. So it's free for the educational use. So you can download the version for the education use, and then you can use it freely. All right. So far, so good. Now we already, as I discussed with you guys while we were looking at the web page of our UniProt page of our protein, that our enzyme of interest, the enzyme alkaline thermostable alkaline protease, for which we are looking whether it can be a cheap alternate for the 7-ase, the structure, the NMR or experimental structure is not available. The only thing available is alpha fold structure, which is also a prediction, but we will also be predicting our structure, a structure of this enzyme using basically Swiss modeler. So now we will move on to that step from here. So I'll just going to close few windows here. All right, so that we can directly go to our page. Okay, here we are. So now let's go to the UniProt Swiss modeler page. So you can search this in the Google. It will provide you the link as well. Here we are. Okay. So again, so we want to use this Swiss model. Okay. Here you can use the accession number of your protein here, or you can put in your sequence. So this is a sequence of our alkaline protease enzyme. So I'm going to copy this sequence here. So this, it has actually identified that, yeah, this template, this sequence is a template, and we can actually go for the structure of model of this enzyme. But let's just see what this Swiss model, which provide us a homology model of our enzyme. I'm sorry. But what is homology modeling? I discussed earlier. We'll come back to this phase for this homology modeling thing later on. So basically, in the homology model, what we know is we only have the sequence of our protein, and we don't have any structure. But in the database, someone has solved the structure of a protein which is very similar, which has a very similar sequence, around more than 60% sequence identity or similarity with our protein, and whose structure is available. So we can use that structure to build a model of our protein structure. So for example, this is an unknown structure. This is a protein whose structure is unknown, so we don't know the structure, and only thing we available to us is its sequence. And this is our template. Template is a protein which is has a pretty good similarity or homology to our protein, and its sequence and structure both are available. So we can use its structure and sequence to build a model of our protein structure. So this is basically homology modeling. Okay, so we are going to use this. So what Swiss modeler is going to do now for us, it will actually start looking for a template. So in the database, once we have put in our sequence, it will actually start looking, is there any protein whose structure and sequence is available and it is similar to our protein. It is a huge homology or similarity to our protein. Is there any such protein available? If yes, it will provide you a list of those proteins, and we can select one of those proteins to use as a template to build a model of our protein. Okay, so this is what we are going to do now. Homology modeling is basically a way where you have the sequence of one protein and a sequence of a template. Sequence and templates and structure of the template are available. It will first align both sequences based upon their similarities, and then it will use the structural features of the template to build a model of your protein of interest. Okay, so it will first do this alignment, a pairwise alignment with your sequence, and then later on it will use the structural features of template to build a model of structural model of your protein of interest whose structure was not available. And this is basically the end of my presentation. So let's build the homology model of homology based model of our proteins. So we can give this a title, for example, alkaline protease, and I can give it my email ID. You guys can also note down this email ID, and if you have any question later on, you can send it to me. You can email me or your feedback on this tutorial if you want. Okay, so let's you can directly build model. If you click on the build model, it will not show you what template it is you're going to use. It will look for the templates, and whatever it finds best, it will use that template to build a model and give you a model in one step. But it's better that if we first search the template, look at the templates it has actually come up with, and then we go for the model building. So let's search for the template first. It'll take a while. Let me just go back and see if anyone else has any question. All right, so no questions so far. Okay, I hope it has come up. No, it's still searching for the template. Let's wait for a while. Okay. All right, I hope it come up with some templates. So it's going into the database, looking for trying to align our sequence with the templates available, and see if if some of them are good enough for model building. it is showing what kind of stuff it is doing here, how it is actually searching for the template at different steps involved. We're not going into that detail, but here we are with all the different templates it has searched for us. All right, so these are basically the structures of the enzymes available and whose sequences are pretty similar to our proteins. So first one, it has the one at the top is the one it thinks is the best based upon different scores and coverage. The coverage mean how much of this structure available in the database covers our sequence. So it almost covers all of it. Only I believe there is some additional part available in the model or in our sequence. So we can have a look later on. Then it has this global model quality estimate, which is highest for the top sequence. Then it has some quaternary structural quality assessment, which is not available at this moment because it has not built any structure so far. Sequence identity has been given, so that one at the top has almost 49% sequence identity to our sequence, our protein of interest. Then the method for this model, the template the structure has been solved using X-rays. It's a monomer, it has only one chain, it has a ligand attached to it, which is calcium. There are other sequences available. Some of them have even more sequence identity than to our protein, but it has less coverage. So a part of our sequence is not present in this template. That's why it's global GMQS score is low. It's better to choose the one which has the highest coverage and highest GMQE number. Um, if you are just a beginner, if you just want to build a model, but if you have some specific research reason or a question or some information or some data available based upon some literature studies. So you can select some other template as well, but for that you need to have a good reason why you are selecting them. So for today's, we are going to select the one which database think, Swiss model think is best based upon GMQE and coverage. Right. And then we'll go and look from other structures. The quaternary structure of this protein, the template has been given here. Then the sequence similarity, how much similar this sequence is with our sequence has also been, some details are given in here. Alignment. So here it has aligned our sequence with the template. So this is our target. So this is our sequence. At the top, it is our sequence, and in the second line actually represent the template, which is we are going to use information from our model building. So you can see only this N-terminal small region has not been aligned, and there are some bits which somewhere here in the sequence which is not present, which didn't align. There are some gaps, but mostly the structure covers almost all. The template covers almost all the target. Okay, so I believe it's good to go. So let's build a model. Right. It also takes a while. I'll go back to this main page to see if someone else has any question. What is the highest GMQE value? It could, yeah, it could go to one because if there is 100% similarity, then it will go to one, and coverage as well. So it means that you are using the same sequence or a very similar kind of a so same sequence for building the same structure. So you are using a structure, a sequence whose structure was already available. So it will give you one. That is highest similarity quality assessment. So it goes from 0 to 1. Someone answered this question already. Thank you. All right, let's go. Any other question? Any other question? Let me see. Oh God, it just moves so quickly. The sequence which is pasted on Swiss model page is of an unknown protein. Yeah, it is of our protein. So here we were. So we had two proteins. So this 7-ase enzyme, which is commercially available, its structure is already available. So the enzyme we are industry to replace it is the one which is alkaline, thermostable alkaline protease. Its structure is not available. So we pasted the sequence of this enzyme in the Swiss model. So its structure is not available. So we are building a model of this enzyme. Okay, because we wanted to know, we wanted to look at the active site of this enzyme. We want to compare the structure with the 7-ase structure which is already available. The crystal structure of 7-ase is available, so we would not build the structure of 7-ase, but it's this enzyme, this enzyme of our interest, its structure is not available. So that's why we are building this structure, and we use the sequence of this thermostable alkaline protease to build the model. So that is what we pasted in the Swiss model. Okay, right. Hope, Sabah, I have answered your question. All right. So here we are with our model. Okay. So okay. So here they have actually this. This is our model. Okay. You can go around and you can have a look on this one. Okay. And then using this page, you can actually come back to the normal. So we can. I'm sorry. Let's. We can assess our sequence. For example, here an alignment of our sequence with the target has been given in detail with the secondary structures highlighted. Arrow represent the alpha helices, and these bundles or barrel shapes, these represent the beta sheets. So you can click any one of these residues and it will be highlighted on the sequence in this window basically. Okay. But for the assessment, the quality of our structure or our model, we can go and see here. So we can go the structure assessment. Okay, let's go to the structure assessment. And structure assessment would give you a Ramachandran plot for your model. And here you can see most of, so the green bits are, the darker it is, that more favorable it is. So each one of these dots basically represent a residue of your protein. Okay, and the in Ramachandran plot, if you know the Ramachandran plot already, these are basically phi and psi angles, which are and these green actually areas represent the allowed or favorable regions, allowed regions of Ramachandran plot for any protein structure. So what we want for our model, that all that, so each residue, all these residues should be in the allowed regions. So as you can see, apart from a slight, which is a valine, very few residue. This one is little slightly off. They're outside in the white areas, but most of them are in the green regions. The dark green is more favorable. The light green, as you go lighter, they are slightly bit uh, favorable, less favorable in the protein structure, but these are all allowed regions. So the green regions are the allowed regions. And for a structure to be accepted, even the structures which are experimentally solved, for them we do the analysis on Ramachandran plot, and we only accept the structure to be correct or true if all the residues have these phi and psi angles in the allowed regions of Ramachandran plot. So you can see it's kind of a good model because most of its residues are even in the dark green regions of Ramachandran plot. Later on, it gives you this mole probability results provide you that 93% features are in the allowed regions. There are only 1.5% outliers. Some are rotamer outliers, some have some other deviations, beta, C beta. But mostly 93% regions are in allowed regions. Some are bad angles, but and there are some steric hindrances and clashes available. So they have given the detail, but we are happy that 93%, more than 90% of our residues are in the allowed region. So that means it's a good structure. Then it has also given to this G mean this. So it has also been given you the score. Basically, you can click on this one and predicted local similarity to the target. Okay, so in most of the region where alignment was good, it is above the the chart is going above zero point 0.8, and where the alignments were not so good, for example in N-terminal regions, it's a bit lower than zero point 0.8. Okay, so this is basically quality assessment, and these are on x-axis we have residue numbers. Right, so this q mean z score is also telling. So here you have a lot of deviation, lot of so here is your structure. Basically your model is somewhere here, it's close to the middle or mean score. So it's kind of a good score. If your model is somewhere here or would be somewhere here or here, then it wouldn't be a good model. Right, for this it's an acceptable model. Here you can look at the alignments in detail. So q mean score has been given. So the regions which have more confidence, where the structure has more similarity or has been aligned with the target well, those regions have higher q mean score. That means there is the structure model is more reliable in those regions. And the regions which are next to the gaps, you can see whether the q mean score is low, and because the homology was in it coming, it is coming after the gap, so there was less confident way for the structure which has been provided here. uh, but overall in most of the regions it has a good q mean score. So that means it's an acceptable quality structure. Okay, so what else we can do in here? We can actually go and download the structure from here, and let me go back. Right. Mhm. Oh, I'm sorry. Why the hell I didn't go there? Okay, wait a minute. I hope it will come back. So we have model here. Yes, here we are with our model. Okay, and we can actually download this model in PDB format. Okay, so now we have our model has also been downloaded. So we built the model. We actually did some assessment how the structure is, and we came to this conclusion that based upon the Ramachandran plot and other data available on the structure assessment, it's an acceptable kind of model. So now we have this protein model available, and what we are going to do in here in the next step, we are going to actually open it with the align it with the structure of our 7-ase enzyme, and we're going to look at the active site of the enzyme. Okay, all right, so I hope all of you are with me. So this window already has our 7-ase enzyme open. Okay, and we can now open our next enzyme, our model in the same window, and then we will align it. So we went to the downloads, and we are going to open. This is our model. So this is our model, and this is our 7-ase enzyme. Okay, you see, so both are available here, and this is the this blue sky blue color or cyan color is basically for the model. The green one is basically our 7-ase enzyme. So our model doesn't have any water residues attached, but it does have a calcium residue, which I can select and I can color it like I already colored the orange ones in the 7A, so I'm going to color it orange here. And what else I want to do in here? Maybe I can color it something else. I can color it something like for the comparative analysis, like for example, white. So I'll color this. So but it has only one calcium attached to it. Now we actually want to compare these two structures. How we can compare these two structures? What we can do, we can align these structures. So this is our model, alkaline phosphate. I can rename this instead of, I can align it. Let's keep it as a model so that you know this is alkaline model, but I can rename it a little bit to, I'm sorry, this is a bit annoying that it's not working very well. Right. So let's align this one. So you can actually align these residues. So we want to align it to our 7A structure. So here we are with the 7A enzyme. So you can go to action, you can select align to the molecule and then select the other structure available, which is 7A enzyme structure. Sorry. Give me a moment, please. Okay, here we are. So now our structure has been aligned. So you can see, look at the structure and you can see most of the regions, the structure, the alpha helices and beta sheets have been aligned, but our model has this N-terminal extra structure which was absent in our 7A structure. So that's why it didn't align. And if you look at the sequences have also been aligned. So let's highlight the active site of our model. So this is D residue, this is histidine. Oh, no, it's just the active site. And then this is the serine residue. Okay. So we are going to highlight the active site of the model. So this one, we are going to rename it as active site. And active site, I would like to rename it as model active site. So that I know that it's not the 7A's, it's the model we built. It's active site of the model we built. All right, now we have the active site of our model. What we want is we want to color it differently. For the 7A's, we give it red color. For this, I can give it a blue color. And then I want to show it in the form of sticks. And so let's go and have a closer look. So using the right mouse, you can actually zoom in and then you can look at the um, you can position this by pressing the central button of the mouse and then scroll down button, and then from the left you can go up and down. So for PyMol, you would need a mouse. You cannot use it without the mouse on your laptop. It's better to use a mouse so you can position. So you can see that there's a pretty good similarity in the orientation of the active site residues between the two structures. So that also give us a confidence that our model is of a pretty good quality. And also because the enzymes have very similar structures now, and they both are serine proteases, their active site residues are aligned very similarly. So yes, we can actually give it a go. We can use our enzyme alkaline thermostable alkaline protease as an alternate. We can give it a try as an alternate of 7As, and it will actually save a lot of money because it's not a commercial product. It can actually be a cost effective alternate for our if instead of 7As. So we can based upon the sequence analysis and structure analysis, sorry, the active site analysis, we can we can say that yes, we can answer this question to be yes, because our structural and sequence analysis gave us a gave us quite similar kind of results, that the proteins are quite related. Either we can go in the lab now and we can perform a few more experiments with the confidence from our computational analysis that it will be a good investment to try thermostable alkaline protease to replace for Saviti's alternate, right? So yes, so this is basically the end of our tutorial session, and I'm open for the questions. If you have any question, please write it here, and I can answer that. Anything you want to repeat, anything you want to know more, or any comment you want to give, you can write in the chat box, and I'll answer your questions. If all good, then how the Sabah, maybe you have joined later, but Uniprot KB, you can go to this web page. So you can actually let me tell you, you can go to Google. Let me just repeat this, but it was there in the beginning of uh, our tutorial. So Uniprot KB is a protein sequence database. So you can go to this one. You can write down a session number of your protein. If you have the session number, a unique identifier for the sequence of your protein, you can write that down. But if you don't have the session number, you can write down the name of your protein. For example, in my case, it's thermostable alkaline protease. I can write down the name of my protein here and search for it. So it's asking me whether you want a table or you want a card. So I want it to be in a tabular form. So here it has a list of enzymes with the proteins sequences available with the bit more detail, the gene name, the session number. Here's a name. So this is what I'm looking for. Then this is the organism. So I know my protein belongs to the length of the protein. So you must know something about your protein. You know its name, at least you would know uh, which organism it belong, maybe length of the protein. So using that information, you can select the protein of your interest. So I know that my protein is alkaline, thermostable alkaline protease, and based I did some earlier study, and I know that it's coming for this organism. So yes, I'm confident this is my protein. So then I click on this session number. So here we are. If I'm confident this is my protein, if not, then you can actually go down and look for the other details, other organisms, the proteins coming from other organisms, and you can choose the one you are interested in. So then you can go to the session number. Here it has all the details. It has the features, it has structure linked to the structural databases, post-translational modifications, cofactor, the function of the protein, the organism has been given. Then you can click on the sequence tab and you can actually download the sequence from here. So this is a sequence of your protein. So you can take the sequence of your protein of interest from Uniprot KB. Okay, it's a protein sequence database. I hope I answered your question. Do we need to validate in the lab after getting the results from computation? Mostly, yes. We do validate the data from the commercial. If we see, for example, we don't want a setback. So at a pilot scale or at a small scale, we will validate our results. Okay, I do you want me to repeat the aligning step? Okay, since I have already done the alignment, so it wouldn't do it again. But this is my model. Okay, the blue one is the model, and this green one is Savina's enzyme. So you can go to the model action. On the action click, here is an alignment tab. In the alignment tab, you go to the molecule. So we are not selecting any active site or anything. We want to model align the whole molecule, our model, the protein sequence with our Savinase enzyme. So this is a Savinase PDB ID, which is already opened in the same window. So you have to open the structures you are aligning in the same window of the primer. Then under the actions, you can use this alignment tab to align the two sequences. So this would align the two structures as well as the sequences. Okay, I hope it's clear now. All right. Okay, anyone else has anything to say? All right, what way is it to judge the quality? So here is, it depends what you are interested in. For our tutorial, we were interested, we focused on the active site. Okay, so that because we focused on the active sites, so that's why we align the sequences structures, and then we actually looked at the active site of the two enzymes. So the residues, the protein protease active site residues, we look at their alignment, whether they align, how they are oriented, whether they have oriented in a very same kind of directions, 3D coordinates are same. So we actually assessed from the alignment the active site. Uh, for other purposes, for your research interest, it might be the binding site, it might be the whole structure. So that's why it depends upon your research question. But for this tutorial, from the alignment, we focused on the active site, and then we also looked at the overall structure. So you have seen here that the overall structure also align very well. These alpha helices, these beta sheets, although some of these loops and turns are not aligned very well, but that's acceptable because loops are usually the moving bits in a structure. We focus more on these secondary structures and their alignment. So you can see a lot of alpha helices and beta sheets are aligned quite well in these structures. So, and then the active sites are very oriented in a very similar manner. So that's these are the things we focused from the alignment in this tutorial, to assess whether we can actually use the alternate enzyme. Yeah, it does. Yes, you can actually get the numerical coordinates. That's not the scope of this presentation, but yes, you can actually get the numerical coordinates for the active site residues or any residues you are interested in here. For example, you can look at it and you can go and get the details even from the structure and the sequence point of view. You can get the numerical coordinates. It's usually present in the PDB file. So if you're interested in the numerical coordinates in XYZ plane of your or more detailed kind of analysis, you can open these structure files in Notepad and Then for each residue, in front of each residues, its XYZ coordinates would have been given, and you can look at those, right? Okay, I guess this is basically so their similarity will ensure the proper model. No. So similarity for the for the model assessment, because we didn't use 7As as a template. For the model assessment, we did the model assessment here on the on Swiss modeler. So for the structure, whether our model is acceptable or not, we do use this Ramachandran plot. We use the data here. We use this skew mean. We use this alignment. So you have to look at this very, if there is a lot of gaps here, and the alpha helices and beta sheets of your structure and the template are not matching properly in most of the regions, then you have to reject the model. Okay, here from this assignment. In the PyMol, what we did, we didn't use 7As was not our template. So 7As would never ever tell whether how good our model is. 7As, we align our model with the 7As just to answer our question whether we can replace 7As with this thermostable alkaline phosphatase or not. So we have so the 7A structure was already available. The crystal structure was available, already available, but the protease, alkaline protease structure was not available. So we built a model using a different template whose similarity was about 50% with our thermostable protease. We built a model. We used that model here for the structural analysis. So we compared our model with the 7As, and then we looked at the active site and overall alignment of our structure, our model and 7As, and to get more confidence whether we can use alkaline thermostable alkaline protease as an alternate option. So here in PyMol, here by aligning these two structure, we are not assessing the quality of our model. For that you have to use the details and all the information available here on sys model if you want to assess the quality of your model. Okay. Yes, we can. That's what I'm saying, that so far our analysis has told that there is a great chance that we can use thermostable alkaline protease as an alternate. We can do a few lab tests, and then we can just good to go.